Okay, let's have the four levels of protein synthesis. So we have primary, secondary, tertiary, and quaternary protein structures. For the primary proteins, this just refer to the order or sequence in which the amino acids are linked together in a protein. So ito lang yung pagkakasunod-sunod ng mga amino acid na matatagpuan dun sa protein. So, if we talk about the order and sequence, dapat tama yung pagkaka-order nila. Otherwise, this will entail different protein na kapag maiiba na yung sequencing. So, say for example, if the protein structure, say for example, has a peptide of CHEMISTRY, okay, so ano ulit yung letter C? Cysteine, then we have cysteine, and then we have E as glutamic acid, and then we have M as methionine, I for isoleucine, S for serine, T for threonine, and then we have R for arginine, and Y for tyrosine. Okay, so if the sequence is like this, of course, that's a property of one protein. If we are going to jumble it, of course, hindi na yun yung order niya. So, the primary protein structure just refers to the correct ordering or correct order or sequencing of the amino acid. Okay? And for under the primary protein, okay, so this also refers to the zigzag manner or zigzag orientation of the carbon atom and nitrogen atoms that is present in the protein backbone. Okay, so paano ito? So the carbon atom and the nitrogen atoms in the peptide or in the protein rather are arranged in a tetrahedral orientation. That's why they have a zigzag, they are arranged in a zigzag manner. Okay, we have insulin to be the first sequenced protein. It has 51 amino acid, and of course, yung pagkakasunod-sunod ng mga amino acids, dapat tama siya in order for it to be classified as insulin. And we all know that insulin helps facilitate in the conversion of glucose to the storage polysaccharide glycogen when the blood glucose level becomes too high. And insulin ay ginagamit for those patients that suffers from diabetes mellitus. This is a condition wherein there is a insufficient insulin production or inability to use the insulin produced. That's why they are insulin resistant. Okay. This treatment is done by giving the person insulin via, this should be via subcutaneous injections. Now, kapag hindi makaproduce ng insulin, of course, may mga synthetic, no? We have substitute for human insulin na minobodify nila. The source could either be from the pancreas of the slaughterhouse animal, porcine pig insulin, or bovine cow insulin, and this is used by most diabetics without serious side effects because it contains similar structure to that of the human insulin. Another substitute or resources that were used for substitute for insulin is the genetically engineered bacteria. However, this is very expensive. And we also have insulin obtained from plant, which is the safflower. Okay, so again, um, for proteins with a zigzag manner, arranged in a zigzag manner because of the carbon and nitrogen tetrahedral orientation in the backbone of the protein refers to the primary protein structure. Now, pag sinabi naman nating secondary protein structure, hindi na yung carbon and nitrogen ang involved. Okay? The folding of the proteins is due to the hydrogen bonding between the oxygen atom and the hydrogen atom, or oxygen atom of the peptide linkage and the hydrogen atom of the amino group in other peptide linkages. Okay, so hydrogen bonding, oxygen from the carbonyl rather, and the hydrogen from the amino group. Okay, of the other peptide linkage, and we have two two common types of secondary structure. We have the alpha helix, parang ganito, and we have the beta pleated sheet. Okay. So ulitin natin, kung kanina sa primary structure, the folding or the zigzag orientation or zigzag structure is due to the tetrahedral orientation of the carbon and nitrogen atoms in the backbone of the protein. Pagdating sa secondary, this is due to the hydrogen bonding of, ito daw, the hydrogen bonding, saglit, lakihan natin. Okay. Sorry. The hydrogen bonding between the hydrogen of this nitro group, so yung hydrogen daw na yan, and the oxygen of this carbonyl from the other amino group. Okay? So, meron silang hydrogen bonding between the hydrogen of the amino and oxygen of the carbonyl. But this hydrogen bonding is from different amino acids. So, hindi yung katabi niyang, hindi itong dalawang ito. Okay? Kasi meron tayong carbonyl dito at meron tayong amino. Hindi silang dalawa. Rather, yung sa iba, magkaibang carbonyl and amino group ng ibang amino acid. Okay? So, when it comes to alpha helix, so this is the structure. So, parang helical siya. So, this happens in a single protein. So, meron siyang coil configuration. And this coiling or folding of this strand or of this chain is due to the hydrogen bonding that is present in the intramolecular reaction. So, intramolecular kasi nga yung kanyang hydrogen bonding is within the chain itself. Hindi kailangan ng ibang chain. Hindi siya intermolecular. Intra meaning within its chain. Okay? So, ulitin natin. For the secondary, the reaction or the hydrogen bonding between the hydrogen and oxygen, that hydrogen is from the amino group of one amino acid, and the oxygen from the carbonyl group of another amino acid. For the beta pleated sheet, okay, so makikita natin parang meron pa rin siyang zigzag manner here, and that's the primary structure. However, for beta pleated sheet, kailangan natin ng two or more fully extended protein chain segments. So, ganun pa rin. The mechanism is, this is the interaction between the hydrogen from the amino group and oxygen from the carbonyl group of different amino acid residues or amino acids. Okay? So, ganito naman yung ating beta-pleated sheet. Nevertheless, ang dahilan kung bakit merong folding dito at merong folding and coiling ng mga ng mga proteins is due to the hydrogen bonding. Okay? We also have unstructured segments. This is neither helical or beta-pleated sheet. Okay, so sa isang chain ng protein, pwedeng meron tayong beta-pleated sheet, meron din tayong alpha helix, and meron din naman yung mga unstructured segments. Okay? So ito, meron ding alpha helix. So depende sa interactions ng kanilang mga molecules. Okay? So, another structure is the tertiary structure. So, recap natin. Kanina, dun sa primary, is just the sequencing or ordering of the amino acids. Meron tayong zigzag structure because of the tetrahedral orientation of the carbon and nitrogen in the backbone. For secondary naman, the folding and coiling, the alpha helix and the beta pleated sheet happens because of the hydrogen bonding between the carbon of the because of the hydrogen in the amino group and the oxygen of the in the carbonyl group of two different amino acid within the protein itself. Now, pagdating naman sa tertiary structure, the folding and coiling in the protein is due to the interactions between the amino acid side chains or the residue or the R group. Okay, so remember that the R group is just the uh, identity of the identity ng isang amino acid. Kasi nga, parehas naman silang merong, okay, i-recap natin kung ano ba yung R group. For an amino acid, it has the alpha carbon atom, palagi siyang merong amino group, tapos meron din siyang carboxyl group, the COOH, and then meron din siyang hydrogen, and ang nagbabago lang sa kanya ay yung R group or that is the side chain. Okay? So, sa tertiary structure, yung interaction naman ngayon ng mga R group sa isa't isa ang dahilan kung bakit may folding and coiling within the protein structure, and that will fall under the tertiary protein structure. Okay? So, let's have the four interaction responsible for the tertiary structure. So let's have covalent disulfide. So this is the strongest interaction. This results from the interaction of the sulfhydryl or the SH group of 2 cysteine. It's either intermolecular or intramolecular. So kapag merong dalawang residue na cysteine, okay, magkakaroon ng attraction. So, may folding dyan at may coiling. So, paano to? Yan. Ito. May dalawang cysteine daw tayo dito. So, magkakaroon sila ng disulfide linkages or covalent disulfide bond. Okay. Ito yung ating backbone ng ating protein. Meron tayong side chain dito or residue, amino acid residue. Hindi naman sila magkasunod, 'di ba? Pero dahil sa meron tayong cysteine group dito and meron another cysteine group diyan, magkakaroon sila ng interaction. So, with that interaction or disulfide bond, covalent disulfide bond or disulfide linkage, magkakaroon sila ng interaction. So, mafo-fold yung ating protein. And that is the tertiary structure. Okay? Then we also have electrostatic attraction. Paano naman itong electrostatic attractions? This is the result from the interaction of the acidic side chain or R group and the basic side chain. So, i-recall natin, ano ba yung mga acidic amino acids? We have aspartic acid and glutamic acid. And then for the basic, we have histidine, lysine, and arginine. Okay? So, for this electrostatic attraction, so ito yon yung ionic bond niya. Okay? So, this part so ito yung side chain ng ating, I think this is lysine. Okay? Lysine is a basic amino acid. And then, we have here on the opposite of that, yung nagka-interaction na niya, we have here the glutamic acid. So this is base and this is acid, and this is a basic side residue rather, amino acid residue. So meron na naman silang pooling dyan. So may interaction sila dyan. And that is for the ionic bond or electrostatic attraction. Kaya, ayan, nagkaroon ng interaction and hence mayroon ng folding dyan sa dalawang yan. Okay. Meron din tayong tinatawag na hydrogen bond. So, kanina yung hydrogen bond natin sa secondary structure is because of the interaction of hydrogen and oxygen. Hydrogen from the amino group and oxygen from the carbonyl of the peptide linkage. Pero dito sa tertiary, ang hydrogen bond natin is because of the interaction of two polar side chain. Okay? Two polar side chain. Ito naman siya. Example nito. Okay? So, we have here, ano ba yung polar dito? This is actually a serine that is polar neutral. And then we have asparagine. So, serine and asparagine are both polar. So, nagkaroon ng folding dito sa ating peptide or sa ating protein structure because of the interaction of these two polar amino acid residue. And that is due to the hydrogen bond. And the last interaction is kung kanina two polar, ngayon naman ay two non-polar. And that is the hydrophobic attraction. Okay? So, non-polar naman. Non-polar, magkakaroon sila ng interaction. So, this is valine and another valine. Okay? So, parehas silang non-polar. Okay? So, nagkaroon ng folding dito because of the hydrophobic interaction of two non-polar amino acid residue. Okay? So, ulitin natin for the primary, this is just the sequencing. Meron tayong zigzag manner because of the tetrahedral orientation of the carbon and nitrogen in the backbone. Secondary, because of the hydrogen bonding of the oxygen of the carbonyl and nitrogen in the amino acid in the peptide linkages of two different amino acid in the protein. And then for the third structure is because of the interaction of the side chain this time. And if we're going to have two cysteine as a side chain, we have covalent disulfide. If we have basic and acidic side chain, we have electrostatic attraction. If we have two polar, we have hydrogen. And if we have two nonpolar, we have hydrophobic attraction. Okay? And the last is the quaternary protein structure. This is just the interaction of multimeric. Ulitin natin, ano yung multimeric? Merong more than one chain of protein. So, for this example is hemoglobin. So, ayan, meron siyang apat na different chain of proteins. Nevertheless, meron pa rin siyang mga alpha chain dyan, meron pa rin mga beta chain. Okay? So, that's the four structure of proteins.